Morning everybody, welcome to another edition of the Lab 207 webcast. My name is Mr. Kite. I'll be hanging out with you today as we continue on in our series about DNA. The topic for the day is going to be translation. Forgive me, the last couple videos have been kind of long, so we're going to be a little quicker today. Our objectives, things you need to know by the end of the video, are as follows. First of all, understand the purpose of translation. Why do we have it? Why do we do it? What's it about? Second thing, describe the process of translation. So pretty easy. What is it? How's it done? Let's talk about it. So first, the why. So we've talked about the fact that DNA is a giant molecule, but DNA has got all of the information for everything that goes on in the body. DNA is trapped in the nucleus. In the video about transcription, we talked about how that DNA is transcribed into a molecule of mRNA, messenger RNA. That messenger RNA carries the message out to the cytoplasm, but we still have not gotten anything useful yet, because it's kind of like we just took a big book, transcribed it into a small book, passed that small book out into the cytoplasm, but that isn't any working machinery that can do anything. So we've got to make that small book into an actual protein, because it's the protein that does the work. So the whole purpose of translation is to take that information off of the mRNA and then actually transcribe it into a functioning protein molecule that can do some work. Now, scratch what I just said. The mRNA itself does not become the protein. It is just the instructions for building the protein. Two pieces of vocabulary that are going to be really important as we discuss this: codon versus anticodon. In our string of mRNA that we built during transcription, we took our DNA, so let's say our DNA template code was ACTGCT. The matching mRNA transcript would be UGACGA. Now, the way our body reads this, or the way the machinery in your cell reads this, is mRNA is broken up into 3 base sections. Each 3 base section of the mRNA is known as the codon, and ribosomes read the mRNA 1 codon at a time, so 3 base sets. Now, as this process of translation is happening, tRNA is going to be coming in and matching up with these codons. tRNA, this is tRNA right here, or this is part of tRNA right here, has a little end on it that's kind of like a plug. That plug has got a set of 3 bases called the anticodon. The anticodon matches up with the codon. So this right here would be our mRNA. So the mRNA has got a codon that is U, C, C or U. We'll talk about that wobble here in a second. But 3 bases. The tRNA that's coming in has an anticodon that matches those 3 bases. So it's kind of the way that the mRNA signals out and says, hey, this is the tRNA that I need to come and bring this chemical along with you. So this whole process of transcription and translation is centered on that base pairing that we had talked about previously. All right, so the players that you need to be aware of in this process: you've got mRNA, which we've talked about. That's the messenger RNA that is built off of the DNA. It flows out into the cytoplasm. You've got the ribosome. The ribosome binds onto that mRNA. The ribosome is actually going to do the work of reading the mRNA and building the protein. You've got tRNA. tRNA is responsible for bringing the individual amino acids that are going to be built into this growing peptide. And then I got rRNA, which is ribosomal RNA. That is the RNA that makes up the ribosome that actually reads the mRNA. Amino acids, those are the building blocks of the protein that will be growing out of the top of our ribosome. All right, let's talk about the process. You always got to know where to start. So for our process of building a peptide chain, the ribosome always binds onto the start codon. The start codon is a set of 3 bases, and you can kind of see them here in the picture. The start codon is AUG right there. And AUG tells the tRNA that carries methionine, which is an amino acid, to come over. So this tRNA has an anticodon of UAC. They match up. This is the first amino acid in every protein right there, is methionine. So ribosome binds onto the start codon. That's where we're going to begin the process. From there, we've actually got to start making our protein. And the way this is going to work is essentially the ribosome cruises along our mRNA strand. As it cruises along the mRNA strand, tRNAs come in. Each tRNA has an anticodon that matches the codon on the mRNA. Each tRNA is also carrying an amino acid. There are 20 different amino acids. Now note that there are more tRNAs than there are amino acids, but each tRNA is going to bring a specific amino acid. That amino acid will be added to the chain, and then the tRNA will leave and go get another amino acid and bring it over. You see down there in the note that ribosomes have an A, a P, and an E dock. Here's what I mean by that. So in our ribosome, you can see that it is made out of two pieces. You have got the small subunit right here, which rides along the bottom of the RNA. You have got the large subunit right here. Inside this ribosome, you've got right here's the A site, here's the P site, and then over here there's one they're not showing called the E site. And the way this works is the ribosome cruises down our mRNA one codon at a time. So this A site is the site that is open, and let's say that this A site had a codon in it that was, I don't know, C, C, G. That open spot tells the cell that the tRNA with the anticodon that matches this, so it would be GGC, needs to come in, and when he comes in, he brings his amino acid with him. He'll bind onto that site. Next to him is a neighbor that is carrying the growing polypeptide chain, and E is the exit site. So what will happen is when this guy binds down, the ribosome will scooch over one codon. When he scoots over, the tRNA that has landed here will pass his amino acid over to this chain right here that is growing. That is our polypeptide that is being built. And also as the ribosome scoots over, the tRNA that was right here gets bumped out and he exits to go get another amino acid to come back in. So this whole process of building a polypeptide is just done by this ribosome scooting along one codon at a time. Every time it scooches over, it makes a new open A site that the tRNA can drop into. Once that thing drops into the A site, it passes its amino acid over to the tRNA that's in the P site, and then as the ribosome cruises along, the one that's in the P site goes to the E site and is kicked out the back. And like I mentioned just a second ago, each tRNA carries a specific amino acid, so the tRNAs are labeled by their anticodon, which you can see down there at the bottom, and they've each got their own piece of the puzzle that they bring along to add to our growing polypeptide chain. And all of those amino acids, as I've talked about previously, are the building blocks of our peptides. So every tRNA brings in an amino acid. Those amino acids are added into the protein, just kind of like letters in an alphabet, and obviously the order of amino acids is going to determine the shape and the function of our growing peptide chain. And you got to stop somewhere. So the way that this works is, you can see up there at the top, the ribosome is cruising down the mRNA. When it gets to a stop codon, the stop codons are UAG, UAA, or UGA. When that happens, a release factor is added on, and that essentially tells the system time to disassemble, the ribosome breaks apart, the tRNA goes away, the polypeptide is released to go be folded and processed into its final functioning state, and that is the end of the process. Last thing I want to note on this is your polypeptides or your peptides need to know where they are to go. So as our peptide is being built, the first few amino acids on the front of that peptide are known as signal peptides. And what that is it's a specific set of amino acids that tell the cell this is where this polypeptide needs to go. So if it's being built, you can see down there on the bottom, you got the ribosome cruising down the mRNA. If the peptide, the signal peptide comes out and says, hey, I am going to be transported out of the cell, then a set of molecules called the signal recognition particles will come grab that mRNA and the peptide and the ribosome, carry them all over to, say, the rough ER. They'll bind to the rough ER, and then that peptide will be built into the ER where it can be transported out of the cell. So just know that the first few amino acids are kind of like the signal peptides, and they are saying, hey, this is where I am supposed to go once I am finished. I'm going to take a deep breath because I know that was a lot of stuff. I hope it made sense to you. If not, when you see me in class, let me know. Thanks for joining us on the Lab 207 webcast. My name is Mr. Kite. Hopefully we'll see you again.